In this video, I will go over the main steps, the big picture steps of how to annotate genes in the Pathways project for the Genomics Education Partnership. I want to highlight that there are actually 7 steps, and the first 3 steps that I've highlighted in yellow here are going to be done for you by independent study students. The last 4 steps are the ones that we'll go over in this video. So the first 3 steps, which will be done for you, are to actually locate the place in the genome where our gene of interest lies in our target species. The steps that we go through to do this will be first to examine the neighborhood of genes around that particular gene in our reference species, Drosophila melanogaster. The second step is to locate the genomic region of the target species where that particular sequence lies, and then to examine the genomic neighborhood around that particular gene in our target species. The idea is that not only do we expect there to be conservation of the sequence of protein coding genes, but we also expect the order of the genes on the chromosome to be conserved between related species. So we will use the identity of neighboring genes as further evidence that we have indeed found the right spot in the genome where this particular gene is located. So that is where we're going to start. We've done all of those detailed searches in parts 1 through 3, and we're going to go on to the first item that we'll do in lab. So the first step is to look at and determine what the gene structure is in our reference species, Drosophila melanogaster. To do this, we're going to use a particular program that has been created to detail information about genes in Drosophila melanogaster. It's called the Gene Record Finder. You'll get information about how exactly to get to this online program. So we put in the name of our gene of interest in the search bar at the top, and this is an example of one gene, the PP2A29B gene. What we'll notice here is that the structure of the gene is shown in a diagram format under this RNA details box. And you'll notice that there are a bunch of alternatively spliced isoforms of this particular gene found in Drosophila melanogaster. Down here in the polypeptide details box, we have a table format to indicate which exons are used by which isoforms. I want to highlight that some of these isoforms look very similar to each other. For example, PA, PB, and PC seem to be sharing the exact same set of protein coding exons. This is because they only differ in their untranslated regions. And so since we are only looking at protein coding regions, these 3 isoforms will all produce an identical protein. The second step will be to determine the approximate location of each individual coding exon, or CDS, in the genomic scaffold of your target species. So to do this, we're going to go in, and on this Gene Record Finder page, we can actually get the sequence, the protein sequence, of each coding exon of that gene. So when we went down and clicked on this particular line, a pop-up box showed up with the protein sequence of that exact exon. We'll take that sequence and we will use a blast search to ask where is that particular protein sequence found within the genome sequence of our target species. We will then repeat that for each of the CDSs, or the protein coding exons. At the end of this, we'll have mapped approximately where the protein coding sequence of each exon in melanogaster has its similar sequence in our target species. The next step, after we have approximately mapped the coordinates of each of the CDSs or coding exons, is to go in and very carefully map the location of each CDS. So we'll start off by looking for the start codon and looking for the stop codon. And then we will look carefully at every single exon-intron boundary, and we'll look for splice sites. We'll look for evidence such as RNA-seq data and conservation evidence to determine what is the exact last base of that protein coding exon. We'll repeat that to find the splice acceptor site and all of the additional splice donor and splice acceptor sites. Our overall strategy for this fine mapping step will be to use our exon by exon blast search coordinates from our step 2 to direct you to the exact spot in the genome where the conserved sequence lies. And then once we get there, we will examine the evidence that is displayed in the genome browser. And we will look at different pieces of evidence, including the RNA-seq data, the gene predictor programs, the conservation track of alignment to demyelinogaster proteins, and also the splice junctions, which indicate actual splice sites that were used in that RNA. We will pay attention to the exact base that is the likely last base of the exon. And so here we're coming to the end of an exon, and you see here that RNA peaks drop down, and also there's a predicted splice site right at this location. You see that this is followed by a G and a T, which are the consensus splice donor sites, and therefore the very last base of this particular coding exon is likely this T here, and we would record that exact number. At this point, we would also check the phase of that particular exon-intron boundary. And we would repeat this for all of the splice donor and splice acceptor sites for the entire gene. Once we carefully mapped the location of every single protein coding exon, we will then check our gene model and analyze the results. To do this, we will use a program called the Gene Model Checker, and this is what it looks like. We will enter our species and scaffold name, the name of the gene, and we will enter the coordinates of each of the coding exons. We will then have the model checker check our predicted gene model, and it's important to realize that the gene model checker doesn't know the right answer. This is a real research project where we don't know what the right answer is. So the point of the gene model checker is really to check whether you broke any rules, any basic biology rules, like you told it to look at a place and it's out of the correct reading frame, and so there's lots of stop codons, for example. Or you told it the end of an exon was at a particular place, but there wasn't a splice donor after that. After you test your model, your gene model will either pass or fail, and I'm just showing you what it will look like when it completely passes. If your gene model does not pass the first time, you should go back and try to figure out what you did wrong and try to fix any mistakes that you made on the earlier part. Ultimately, I expect each group to get their gene model to pass. Once it passes, we'll get a fair amount of other data in these other tabs that are shown at the top here, and we'll discuss that detail more in class.